Basically are indicators of long-term trends in the environment. Many of them have life cycles that are a year or longer, so their presence or absence can tell us a whole lot about the health of the system. Well, we're fortunate, as Matt mentioned earlier on the fish study, that we have some pre-fire and pre-flood data. And so what I'm going to be doing then is comparing the post-burn flooding with pre-burn data, and we have a summer sample from June 2007 and a fall sample from 2007, which will then become the references for these comparisons. And what I did in this is compare summer to summer to summer, and then fall to fall to fall. That becomes our reference. So we have 2 references and then 4 posts. I'm bringing this up. This is a slide from a graduate student's paper, and the word study appears in here. We'll call it a project area. But this is the Toledo area that was burned earlier in November of 2005. And you've seen the pictures of the fire that came through the catastrophic fire. Came right in through here, hit the upper Indios, and then there was a section on the upper end that was not burned. So that could also be used as a reference. I've taken samples there, but I will not be comparing them today. But as Matt showed you, there were some sites that were established in the upper meadow of Valle Toledo. And then we have those Indios, and then we have some sites here on the lower, the junction of the two, the San Antonio and the Indios. These are a little bit different sites. The Indios coming in from the side, it's a little higher gradient, and at the time that we did our study in 2007, it was about a foot deep and a foot wide. It had some nice undercut bank with a very nice little stream meanders through there. And the Toledo meadow was maybe a couple of feet wide and maybe a few inches deep as far as the water. It had a lot of vascular plants in it. So it was a little broader and shallower. And then the area downstream here was even still wider, shallower, but it did have undercut banks, quite a nice meadow meander situation. We did do some sampling a little further downstream in one of the exposures. So what we did was take some quantitative samples of the macroinvertebrates, and this is not a complete faunistic study for that area. It's just a sample of the riffle-run macroinvertebrates, the more common organisms in the system. So it's not a complete taxa list that we generated, but we've collected close to 100 different kinds of macroinvertebrates in this study. So you take your quantitative samples, you keep your samples separate, store them, preserve them, and then you have to go back to the laboratory and do the processing. That's why you see an old man standing up here. We don't have a lot of young people that are interested in sorting samples. You have to go in and sample, pull the organisms from the debris. So this is sort of a tedious, time-consuming process, but it's something that has to be done. So we try to identify the organisms as low as we can to operational taxonomic units, sometimes down to species, but primarily general. And what we did were some exploratory analyses, that is box plots and analysis of variance on comparing the 2 pre-burn samples to the post-burn. And usually what people want to know is what's the density of the organisms, how many are there per unit area, and then how many different kinds of organisms do you have, and that's where you usually just stop. But we've kind of carried this a little bit further now. We're getting into what we call multi-metric analyses. That is not just how many are there and what different kinds are there, but we start looking at the community as a whole. That is, what are the tolerances of these organisms? Are some of them sensitive or some of them tolerant? We look at EPT, which is mayflies, stoneflies, and caddisflies. These are the 3 and say that if you have a high number of these organisms in your system, you have a fairly clean water, cold, clean water environment. We then look at the relationships of the mayflies, stoneflies, and caddisflies to the chironomids that are part of the system that give it balance. We look at community loss. What is the similarity from a reference site to a impacted site? Is there a group that dominates this? How does that affect diversity index? If you have one group in there that's representing 50% of the organisms, your diversity is not going to be very high in that community. And then we look at the functional feeding groups, the scrapers to the filter collectors, the shredders to the total. So this is sort of a more complete way of looking at the whole system, not just how many and what different kinds. We look at the community, sensitivity, the function, etc. Okay, you use a circular type sampler. This is a quantitative sampler. If you say to yourself, hey, that looks like a 5-gallon paint bucket, well, that's what it is. This is a modified circular sampler, and it's a small sampler for a small environment. So we just go in at those locations, take a sample, end up with a collection of debris, a few stoneflies in here, and then you just pick the specimens out, count them, identify them, and then you can perform the metrics on it. Now, I just mentioned on the metric calculation is that you're going to use a reference site, and we developed metrics for the whole state of New Mexico based on regional metrics, but working in these small systems, we can then either use a site that was unimpacted above the area, or we can use our pre-treatment data, and that's what we're doing then. So we're comparing post to pre. Well, this is one of the main players in the scheme here. This is Hydrocyte. This is one of the caddisflies that is, we call it a collector filter because it makes a filter net, and it catches material that moves downstream, the diatoms and debris and so on. So it is a collector filter, and this is one of the more dominant organisms in the system, or it was, but it is coming back in some of the areas, and I'll make a few more comments about that later. And then this is the beaded. This is a collector also, but it's a gatherer. It just kind of goes out and runs around the environment and scoops up debris and particulate matter that way. So this is one of the important players too. And then this is Brachycentrus, which is a cased caddisfly. It's the one that you saw earlier. This is an interesting specimen because it is also a collector and a gatherer. Okay. This is what happened after the year at the lower San Antonio site. This is a sewage fungus that came in to the community. The community wasn't hit hard, but Brachycentrus was. Its numbers were knocked down. And I don't know if there was just because of a lot of death and destruction in that system, but we just found a tremendous amount of sewage fungus in that system. So looking at the numbers per meter squared, you can see some low things in the upper valley. The number of taxa, there were some changes, but some of them were not significant. The Rito is perhaps where we had some major changes. And the number of taxa went down just a little bit after the fire. The numbers per meter squared in the lower San Antonio took a pretty big hit in August. And I'll just run through this real quickly here. The number of taxa in San Antonio didn't really go down that much too. So what we're seeing then are some changes, different changes taking place in each one of these stream systems. This is the metric system that we're using here, the multi-metric approach, and we have a scale here above a certain percentage. The impacted site to the reference, it's not impaired, then slightly impaired, and we have moderately impaired, and so on down the line. But if you look at the San Antonio and the Valle Toledo, it took a dip. We didn't have a sample here in August because there hadn't been any flooding. but it dipped down a little bit, but the upper system is starting to show a trend recovery here. But the Rito is the one that really got hit hard. And I showed you the sediment up above. The ash came through, and it was really interesting. When I sampled there in August 1, the ash came through, and it was kind of like, just went over. The critters were still there. But then, as we progressed through the year, the ash and then the sediment that came down filled in that channel, the upper part of the channel. So that it was foot wide and foot deep, became just a couple inches deep and still a foot wide. And the community changed in there. We lost the hydrocytes, we lost the brachycentrus, the beetles were still there, but that system changed physically, and that's what happened there. So I don't know what's going to happen, because that sediment may stay, that large material may stay in that channel for a while. And the San Antonio, below the junction of those two, did take a hit. It recovered, but then a year later, where that bacteria came, or pardon me, that fungi came in, that's kind of troubling. I don't know if that's going to flush out. I'm sure it will flush out. The system is resilient. It's going to clean itself up, but that is still there. So that was rather interesting. So we have 3 systems on the San Antonio, and each one reacted a little bit differently. So it'll be interesting to see what happens when we go back this May, if we get any runoff. Okay, thank you very much.
We've got time for one question, probably.
I was wondering about that sewage fungus. Is that typically in this system and it just bloomed, do you think, or did it come from?
I think there's always something in there. When a critter dies, you know, fungi will attack it, and things are dying.
But this particular fungus then is one that's...
It's there. It's there in the system, but it's not that dominant. But there was so much, I guess you say, dead material in there that it came in. Sometimes you'll see it in a hatchery. You'll see fish, and then once they die, they'll get this fungus on them that will start to break down. So it's something that will probably flush out of the system.
Great. Thanks, Jerry. Appreciate it. All right, our next speaker is Alicia Hodson, our entomologist at the Baez Caldera National Preserve. Alicia comes to us from UNM, and then she did her master's degree at the University of Florida in entomology, and she's just been offered a job with USDA's Systematic Entomology Lab in Beltsville, Maryland, working at the Smithsonian Institution on insects. So this will be her last performance for us here, I think. But Alicia will be talking about the terrestrial invertebrates, which of course form the food base for insectivorous small mammals and birds. So this group is very important, and there's not much known about insect responses to fires in forests and montane grasslands. So this will be really interesting stuff to see what she comes up with here. So, Alicia.
So yeah, I'll be talking about the terrestrial arthropods in the Baez Caldera, the effects of the last conscious fire had on them. So arthropods play a very important role in the ecosystem. They act as detritivores, pollinators, herbivores like grasshoppers and crickets, and also predators. And as Bob mentioned, they're also an important food source to small mammals and birds, and also some larger mammals like penguins and bears also will eat some insects. So when fire happens, the system is affected, and it can either be drastic or not so, but obviously in this case it was very drastic. So in June 26th of 2011, the Lost Conchas fire started just south of the Baez Caldera, and then quickly spread throughout the Jemez Mountain Range, including 30% of the Baez Caldera National Preserve. As a result of this fire, we wanted to look at the effects of the arthropod populations, and we did this in 3 different habitat types: mixed conifer, ponderosa pine, and mountain valley, or as I call it in this slide, it's grasslands. And it has, as it's been mentioned before, the fire provided a nice monitoring testing site of having half of it being controlled and the other half being burned. And in 2011, we deployed 36 pitfall traps, 18 controlled and 18 burned. And then in 2012, we deployed 16 malaise traps to look at flying insects, as well as ultraviolet blacklight traps to look at the moth populations. So the pitfalls looked at the ground-dwelling arthropods, and for those, we did 3 traps per exposure in the 6 replicate stands of ponderosa pine and mixed conifer, burned and unburned. This is obviously a burn site, and operated continuously and collected every 2 weeks. And then the specimens were identified to the lowest levels possible, mostly species. And in the bottom left corner, it's kind of the close-up of what the pitfall sites look like. And then we also did this in the grassland site with the same design. Now for the malaise traps, the case with this is that the insects will hit that black part and then instinctually fly up towards the white area where there's a collecting jar. And for that, we did 1 trap per exposure for 4 replicate stands of ponderosa pine and mixed conifer. We didn't do it for grasslands. And we ran them for 2 weeks each month, and then collected the sample at the end of 2 weeks and identified the specimens to species. And a lot of specimens that were collected here were parasitic wasps, wood-boring beetles, as well as few moths and butterflies. For the UV blacklight traps, there's me and my 2 interns, Eileen and Arden. We did those in 1 trap per exposure for 4 replicates in ponderosa pine and mixed conifer, burned and unburned, monthly for 1 night, and it was collected the next day. And here's just kind of a close-up of what a blacklight trap looks like. There's the blacklight on top connected to a collection bucket where there's a killing agent at the bottom, and this is run on a 12-volt battery. Again, more blacklight collections. And then to see if there was a significant difference between fire treatments of burned and unburned, and if there was a trend over time, we ran a repeated measure ANOVA. And to look at specific times, we ran a means comparison pairwise test. And we only did this for a taxa where there was greater than 10 individuals. So for the grassland sites, we collected a wide range of taxa, going from beetles to grasshoppers, crickets, spiders, harvestmen or daddy longlegs, centipedes, and millipedes. And in the control sites, we collected a total of 6,376 individuals. And in the burned area, we collected 7,336 individuals. Now, some of the taxa from the ANOVA showed that you're either positively affected or negatively affected, and the positively affected by the fire is in the red. And this includes some ground beetles, which are predators within the system, as well as some wood-boring beetles, the serendipity, as well as some plant-feeding beetles were positively affected by the fire. The taxa that were negatively affected by the fire in the green, a lot of grasshoppers were negatively affected in the grassland. And that's because they predominantly live in the grassland. Some other taxa that were positively affected were centipedes and millipedes. And the harvestmen were really negatively affected. There is absolutely no harvestmen in the burn areas, which is really interesting. But that could be because harvestmen normally occur in more of a covered area. They like rocks and being in park in a burned area where there's less of that. You don't think that they would be in there. Here's some graphs just to kind of further support the positive and negative effects of certain taxa. We have grasshoppers on the top two graphs. So they were obviously, there's definitely less in the burned area rather than the control. And the little stars indicate significant time periods where there's a difference between the burned and control areas. And millipedes did better within the burned areas. And it's, you know, it's hard to say. There could be a possibility there's more millipedes within those areas, but it's mostly a trap bias effect because there's less ground cover. They're looking for a place to reside. And here's some predatory ground beetles, as well as that longhorn beetle I mentioned earlier before. And then the some plant feeding beetles also did better in the burned areas rather than the unburned areas. In the mixed conifer group, we had a total of 778 individuals collected in the control and 790 in the burn, which is less than the grassland, but still same kind of wide range of taxa of beetles, grasshoppers, crickets, spiders, mites, centipedes, and millipedes. So the species that were negatively affected by the fire in the mixed conifer, again, is the harvestman. About 2 different species of ground beetles, 2 different species of staphylinid or rope beetles, which are they're fungivores and detritivores. And then the species positively affected by the fire are centipedes, 1 silk and fungus beetle, and cattle crickets. And having the 1 beetle, that fungus beetle, we kind of speculate the reason why there's such a big number of the burned areas versus the unburned is that it feeds on fungus. And so when there's lots of decaying trees and matter within the burn sites, they're attracted to it. And then again, here's the graphs of the predatory beetles. Actually, all of these are predatory ground beetles. We have our rope beetles, our southern fungus beetles, which you can see is mostly in the burned areas. And the cattle crickets, they were affected, but it doesn't seem so much as the fire effect, but more throughout the time. And the other arthropods, which are of the decomposers and predators like spiders and harvestmen, also had a significant effect. And for the results of the moth project that my 2 interns, Eileen and Arden, did, well, this was just for the month of July. There was quite an abundant amount of moths that were collected during last summer. But just in the month of July, 167 individuals were collected. Species. Sorry. And that you can see that there's more in the control sites rather than the burn sites. And again, with the species richness and sample. And highlighted in yellow is the species that were negatively affected by the fire, and you can see it's most of the species, and that's because moths are very flammable. An interesting fact, though, is there's this one species, Phema dovestator, and I looked into the natural history of this species, and it turns out that it's a pest on Kentucky bluegrass, and as Rebecca stated, that after the fire, the Kentucky bluegrass responded positively to the fire, so I think this is correlation to that, that as the Kentucky bluegrass increased, so did its, one of its key pests. An important reason why we monitored the moth populations within the Vice caldera is this key pest species, the western spruce budworm. And here's an overview map going from 2003 to 2012 of the outbreak within the Vice caldera. And as you can see, there's a very large increase within the year 2009. But in 2011 and 2012, you see there is a very large decrease, and that's because of the fire. So we'll be continuing to monitor the western spruce budworm to see if it comes back or not. I would like to mention that the malaise and ponderous pine football tracks are still being processed. As Jacobi mentioned, processing time takes a very long time when it comes to invertebrates. But we're finding a lot of interesting things. Our collaborators at the Systematic Entomology Lab, USDA, who are looking at the malaise samples, have found very large amounts of parasitic and parasitoid wasps within these traps. And those guys are really important because they actually will infect either trees or other insects. So, in summary, there was more individuals collected in the grassland sites than the mixed conifer. And I think, as Rebecca was saying, it's just because the grasslands came back very quickly after the fire, whereas in the mixed conifer, the succession is a bit slower. And then overall, the fire had little impact on ground-dwelling arthropods, but some key taxa or key groups were affected. So like the grasshoppers and crickets, they're negatively affected in both mixed conifer and grasslands. Ground beetles were both positively and negatively affected, and that's for certain taxa. Fungus beetles were positively affected because they're the fungivores and they'll be feeding on the fungus within the burn sites, as well as rogue beetles. Spiders were not really significant. There were a few more spiders within the mixed conifer areas than in the grasslands. And again, centipedes, millipedes positively affected, and harvestmen and moths were negatively affected by the fire. And it's just important, I mean, the arthropod populations are coming back. That's kind of what the graphs were showing as well. They're coming back. They survived on the fire, either through finding refuges within the patches because there was such a mosaic within the fire, or because they had time to, they had wings and they could fly and escape the fire. But it's really important that it's coming back. It's going to be really exciting to see kind of the abundance within the next few years, and just that they're really an important part of this system. So I'd like to thank the Vice Caldera National Preserve, Santa Fe National Forest, University of Mexico, Museum of Southwestern Biology, University of South Suwannee, where my two interns, Eileen and Arden, are from, the USDA Systematic Entomology Lab, not only for their help, but also giving me a job. And of course, the numerous volunteers that helped collect and process these samples. So if there are any questions, I would be.
Time for questions?
Would you maybe be able to correlate moth food plant responses to adult moth abundances? So larval food plants and how they responded to fire?
No, we haven't made any plant insect correlations, but it's just kind of, it was just kind of a circumstance that I was like, oh, hey, here's this one moth and here's this plant. But I think that would be very interesting because if there's a more abundance of a certain plant, or if there's like an outbreak of, say, the spruce budworm moth, that's very important to look at.
Yeah, and I'd just like to throw in, I think as we get further out on this and some of the success discussion goes a little bit further, it'll be a little easier to do that. We can look at the functional groups of the insects that respond to the types of plants that we're seeing. But right now, it's a little early.
Okay.
I would point out that Professor Rob Verblin here from Texas Tech University is the new faculty member there on fire, and she is already analyzing the ant fauna from these various study sites on here. And then we have Dr. Sandra Greenlee at the Museum of Southwestern Biology identifying all the spiders down to species. So we're still not done, we're still probing in here, because when we get results like there's no effect of fire on spiders at the class level of arachnid level, maybe when you get down to the species level, you get all kinds of dynamics. Some go up, some go down. But we don't know that until we can get the taxonomy done. So that's what Sandra's doing. That's what Robin's going to be doing with the ants. So should be great stuff. Other questions? Very good. Thanks, Alicia. Yet another announcement, hot off the press. Please announce that student paper poster evaluations are available for pickup at the registration desk. So during the break, swing over and do that. Get your grades so you know whether you passed or not. My old classes at UNM, we only had 2 grades. They were A's and F's. You're either alive or fatality. So this one, I'm sure they're not quite as harsh here. The wildlife meetings, nature red and tooth and claw. Very good. So, let me see, I'm not supposed to be next here. I'll start introducing Erin here. Dr. Erin Greenlee is another UNM undergraduate that went on to graduate school and got her PhD at Purdue, right? Ohio State. Oh, bad Bob. She's a Buckeye here. And she came back and started working with Hawks Aloft, who is one of the organizations that is collaborating with us on a collaborative forest landscape restoration program. And so Dr. Greenlee has been organizing and carrying out the bird studies, both on the National Forest and the preserve with respect to foresting, prescribed fire, riparian restorations, and so on. But she's also been running the studies on birds from the Las Conchas fire impacts. So without further ado, Erin Greenlee.
Okay, so I'll just get started. Today I'm going to talk to you about our monitoring design, how we monitor bird populations for the entire project, as well as specifically within the caldera. Our habitats and the results. I'll break it down by broad scale results as well as habitat specific results, and then discuss some of those results, and then talk about some of our future goals. What does this play? Well, hopefully it doesn't go through all of them. Okay, so we have 10 project areas spread out equally between the Valles Caldera and the Santa Fe National Forest that's contained within the Jemez Mountains. And our monitoring areas cover 4 different ecosystem types. We have high elevation riparian, mixed conifer, ponderosa pine, and mountain valley grasslands. And a total of 139 point count stations. So let's talk a little bit about those habitats. We have 3 habitat types that I'll be talking about today that we've evaluated in the caldera. So the ponderosa pine, the mixed conifer, and the mountain valley grasslands, which will be referred to now as grasslands. So we have a total of 36 point count stations within those habitat types. And they're broken down by 2 treatment types. We have the burn and not burn, which is kind of serving as a control. Okay, so We don't really need a reminder, but here is one of just how intense the fire was. And the Las Conchas fire served as our treatment for this initial year of the monitoring project. So, familiar map of where the burn occurred here, and looking at the topography map of the caldera. And the main takeaway thing, I think it might be hard to see the details of this map from where you are. But the main thing is to look at these larger circles indicate the different types of habitat. And so this is just the scattering of those point count stations. So here's some pictures of our sites. So you can see that there's a control, the non-burned versus the burned. Here's the mixed conifer and the mountain valley grasslands. And so, before I get into talking to you about the results, I'd like to tell you a little bit of our method of monitoring bird population. So we have a good group of very willing field technicians that get up at the crack of dawn, actually hours before the crack of dawn, to get ready to count birds. And what we do is all our surveys start within 30 minutes of sunrise and end 4 hours later. And we come to a point, we wait 2 minutes, and then we count and record everything we hear and see within a 125-meter radius. We also record anything that extends beyond that 125-meter radius, but the data that we use, that I analyzed here, just was within that 125-meter radius. Okay, so broad scale results. Overall, we documented 72 species within those 3 habitats. The American three-toed woodpecker was one of the species that we expected to show up this past spring, and it did. They really prefer heavily disturbed areas as well as burned areas. And we were able to document what we believe to be the first case, at least known to us, of American three-toed woodpeckers breeding in the caldera. And additionally, we were able to detect significant differences in species richness as well as in abundance. Okay, so here's just a basic graph of the species richness. You can see that in the blue is mixed conifer, in the red is ponderosa pine, and green is the grasslands. And you can see that in the burned plots, there's not much difference between the 2 forest sites, and there's some difference here in the grassland. But the big takeaway difference here is that you can see in the grasslands in the burned versus the grasslands that were not burned, there's a huge difference in total numbers of species that we were able to detect. Interestingly, though, there were a few numbers are different here for the 2 different forest types of the controls. However, those were not statistically significant differences. So because they were not statistically significant, we grouped our forest, our mixed conifer, and our ponderosa pine to just a forest habitat, because we could not find any differences between the 2. So our forest habitats, we found greater abundance in the burned plots overall, but there were several species that stood out the most. We saw significant differences. And it's the American robin, American three-toed woodpecker, and the mountain bluebird. We also found in the forest habitats that there are lower abundance of some species. Among those were the ruby-crowned kinglet and the white-breasted nuthatch. And in the grasslands, we found that species richness, it was really interesting. We found that in the control plots, the areas that were not burned, on average, in total, we calculated we detected 43 species, and it averaged out to be about 48 individuals, so 48 birds per plot. But in the burned, we only detected 16 species, and on average, it's about 18 individuals per plot. And that was quite the drastic difference. So we've also found that there was greater abundance in the control plots, and among those, the most significant were the American crow and the chipping sparrow. So I will get into talking about what some of these mean. So just a summary, we had increased abundance in our bird plots among American robins, American three-toed woodpecker, mountain bluebirds. Decreased abundance among the ruby-crowned kinglets and white-breasted nuthatches. And increased abundance with American crow and the chipping sparrow. And among species richness, we saw decreases species richness in the burned plots and increase of richness in the control plots pertaining specifically to the grasslands here. Okay, so I'd like to stress that this is the first year of first breeding cycle post Los Conchas fire. And it's really critical that I point that out because the response of birds in the initial year will be very different than subsequent years. So habitat structure and food resources change very rapidly post-fire disturbance, and we've seen numerous of examples of that today. It's likely that the initial response of these birds may be mediated by short-term increases in availability of food. So you get increase of seeds or cones or early successional forbs and grasses that move in quickly. And you also see that woodpeckers, they feed on the beetle larvae beneath the bark of the standing dead trees. We also find that it may also be mediated in certain circumstances by short-term decreases in food availability. So in the grasslands, likely there's a reduction in arthropods that the birds were feeding upon, and that would likely lead to a reduction in that probably influence the difference in species richness and abundance that we found in our grasslands. And also architectural differences, so change in standing dead trees, nest site availability, predator coverage. And the architectural differences can apply to both our forest birds and our grassland birds. The short-term decrease of food is likely what is driving some of our foliage gleaning species, so the ruby-crowned kinglet and the white-breasted nuthatch. They typically do not prefer burned areas, and it's likely because there's a reduction in available food and nesting sites for them. So our future goals, these are many, so I just boiled it down to two. Continue monitoring the avian response, but the big thing, the most interesting thing to me is if you look within the literature of birds and bird communities post-fire, these first several years post-fire are often grouped together. And I think it's interesting, and we hope to keep these years separate so that we can watch and see what happens to these birds the first year. We're starting to start the second year this May, and see how populations stabilize, how long it takes for a population to stabilize. With grassland obligate dependent species, it usually takes 3 to 4 years for those populations to reestablish and become stable. And it'll be interesting to see how these species makeup change in both the forest and in the grassland plots. And then it'll be even more interesting to compare that to the other treatment types that will be occurring on some of our plots. And with that, I'd like to thank Bob Parmenter and Joe Wargo. We did a lot of work with her. She's with the U.S. Forest Service in the Ames Ranger District. My 3 stellar field technicians with whom I could not have done this without them. And they were very willing to wake up at 3 in the morning and drive out to the caldera to count birds. Gail Garber is the executive director of Hawkslop. It's just acknowledgement of some of the photos. And with that, take the questions.
did you record burn severity on each of your different plots within
So
those 5?
that's a good question, and I didn't mention that. All of our plots are standard placement severity for, and it definitely does impact difference, how birds respond differently depending on the severity of the bird. And we found that our results line up pretty closely to that we find in the literature for standard placement. Anybody else?
Okay, well, we've got a break coming up, so we get a few extra minutes there. We'll reconvene at the standard time of 10:20, I believe. And Professor Jimmy Kane will be up next, talking about large and small mammals in the post-fire environment. And it's really great stuff, too. I'm looking forward to that. So thank you very much, and we'll see you in 25 minutes or so.